cell division, an essential life process. Now the picture over here shows you how cell is dividing. Cell division is the key to life. From the moment we are first conceived, we are continuously changing and growing. In order for our bodies to grow and develop, they must produce new cells and allow for death of old cells. It is also an essential component of injury repair. If our cells could not divide and create new cells, our bodies could never produce new skin cells to heal road rash, meaning injury to the skin during an accident, or grow a fingernail back. However, when cell division goes awry, dramatic results may occur, and repeated rounds of unregulated cell division can lead to life-threatening diseases like cancer. Cell division occurs by a strict cycle with multiple stages and checkpoints to ensure things don't go awry. Perhaps most importantly, without cell division, no species would be able to reproduce. Life would simply end. There are two types of cell division. Number one, mitosis. Number two, meiosis. Now, before any cell division, the number of chromosomes double. 2n becomes 4n. Now we have got 46 chromosomes, so they become 92. Mitosis is a type of cell division that results in two daughter cells having the same number and kind of chromosome as the parent nucleus. More cells are made genetically same as the parent cell. Mitosis is very important for growth and development of the body. Mitosis is very essential for asexual reproduction. So you've seen over here the doubled chromosomes, that is 4n, have been reduced to 2n. This has been divided, and 4 divided by 2 will give you 2 each. Now the second type of division is meiosis. It is a very special type of cell division. It involves 2 rounds of division. The first round is mitosis. The second round is meiosis. And it results in 4 cells being produced containing half the genetic material. That is 4n by mitotic division became 2n. 2n by meiotic division became n. So you got 1, 2, 3, 4, and they've got exactly half the amount of genetic material. Now, when you come to meiosis, there is reshuffling of the genes, resulting in a great variety of offspring. Without meiosis, we would all look exactly alike. Meiosis allows each offspring to be different and so potentially better than the parent. Now, mitosis only occurs in somatic cells and stem cells. Now, what are somatic cells? Now, somatic cells are the building blocks of our body. Just like bricks form the building blocks of a house, same way the somatic cells form the building blocks of our body. And when we come to stem cells, these cells, they replace damaged cells, lost cells. They can divide again and again and again. Besides, they are very necessary for restoring emaciated body. That is a body which has lost a lot of weight and have become very thin due to the loss of the fatty tissue beneath the skin and muscles throughout the body. It also helps in healing wounds and formation of blood. Now coming to meiosis, where it takes place only in the germ cells. Now these cells are gametes. That is, these are the ones which have the male gamete and the female gamete. The male gamete is called the sperm. The female gamete is called the egg. Now the sperm is present in the testes and the egg is present in the ovaries. Interface. Just before cell division, chromosome number doubles. That means 2n becomes 4n. It happens in a phase called interphase. The cell performs its normal functions and also prepares for the upcoming cell division. During this phase, that is interphase, the cell grows in size. This phase is the longest phase because of the complexity of genetic material duplication, that is, the DNA is copied or replicated. In short, it makes the final preparation for cell division. Throughout interphase, the DNA is semi-condensed form. This cell is in interphase. It has grown in size and the DNA over here is in semi-condensed form. That means it is not tightly packed and it cannot be seen under the microscope. Now if you look at this cell, this is the outermost wall of the cell, which is known as the plasma membrane. The blue liquid what you see is the cytoplasm. Then over here what you see is the nucleus. Inside the nucleus we have this DNA which is now in the form of fibers and it is known as chromatin. They are not yet formed into chromosomes because they are not tightly packed. Then we have the nuclear membrane and we have the centriole. Now you will know the function of them as we proceed. But remember that the nucleus is one which, the nucleolus, it makes ribosomes. And these centrioles are barrel-shaped organelles. That is, they are hollow cylinders which are very close to the nucleus. Now boys, let us see how a chromosome looks once it is tightly packed and it is compact. It looks somewhat like this. Each chromosome has a constriction part which is known as the centromere. It has got two arms. One arm is short and it is called the P arm. The other arm is long and it is known as the Q arm. Now this chromosome has got two identical structures. That is, this structure is identical to this structure and They are called sister chromatids. They are joined at the centromere. The DNA has already been replicated. The final preparation for mitotic phase is completed. Now, the cell is able to enter the first stage of mitosis. The DNA is semi-condensed and is called chromatids. Now coming to the first stage, that is prophase. The semi-condensed chromosomes become more compact and are visible now. They consist of sister chromatids which are paired at the centromere. The centrosomes located near the nucleus are made up of two centrioles which are surrounded by microtubules. Centrioles duplicate and each centriole starts moving towards the opposite poles of a cell. The microtubules which are surrounding the centriole are known as spindle fibers. They extend from the centriole. As prophase progresses, they attach themselves to the chromosomes. The nuclear membrane and the nucleolus start to disappear. Metaphase, that is the second phase of mitosis. The nuclear membrane and the nucleolus disappear completely. Chromosomes are clearly visible as condensation is complete with their sister chromatids. Chromosomes are arranged parallel to the equatorial plane of the cell and remain joined at the central point called the centromere. All the chromosomes are captured by the spindle fibers. These are the spindle fibers. The spindle fibers pull the chromosome back and forth until they align along the equatorial plane. So you can see all the chromosomes are along the equatorial plane. These are the spindle fibers. These are the centrioles which are responsible for making the spindle fibers. And these spindle fibers will keep pulling and pushing the chromosomes till they are all on the equatorial plane. Once the cell establishes that the chromosomes are arranged on the equatorial plane and the spindle fibers are correctly attached to the cell, it enters the next phase. The cell now enters the third phase called anaphase. It is the shortest phase because it involves only the separation of the sister chromatids and their migration. The centromere splits and the sister chromatids of each chromosome separate and are pulled by spindle fibers and reach the opposite poles of the cell. The separated sister chromatids are called the daughter chromosomes. So you can see clearly over here that the chromosome has broken or separated into two pieces and the spindle fibers are pulling them towards their respective poles. So you'll see that this sister chromatid has separated from this sister chromatid. And now what you get over here are known as the daughter chromosomes. The cell has now reached the final phase of mitosis, that is the fourth phase called telophase. Look at the figure while I am reading. A nuclear membrane forms around each set of chromosomes to separate the nuclear DNA from the cytoplasm. So you can see now the nuclear membrane has just started being formed. Chromosomes begin to uncoil, which makes them less compact. They become thread-like, thin and visible and invisible. So these were the chromosomes. Now they have begun uncoiling. They become loose. So now they are becoming very thin and you cannot see them, resulting in two daughter nuclei being formed in a mother cell. The nucleolus appears in each nucleus. The spindle fibers have disappeared completely, meaning end of karyokinesis, that is, nuclear division has ended. And then we come to the last, which is the division of the cytoplasm, and that is known as cytokinesis. The end of karyokinesis means cytokinesis begin. That is, nuclear division is done. Now the cytoplasmic division begins. The cytoplasm of the mother cell physically divides to form two daughter cells, each containing the same number and kind of chromosome as the mother cell. It begins with a notch. Now you can see what is a notch over here, being formed at the equatorial plane of the mother cell, which keeps becoming deeper and deeper, and gradually two cells are formed. In plant cells, there is no notch which will be formed, but a cell plate is formed along the midline of the cell. Cytokinesis is complete.